I'm Dan Schrider. I'm an assistant professor at the University of North Carolina, and I'm going to be sharing results from a little simulation study I've been playing around with you guys. So, probably don't have to convince too many people here that natural selection plays a pretty profound role in shaping patterns of phenotypic diversity, and we could look all across the entire tree of life to see this, or we could just look at leaf mimic katydids, which I think are sufficient to make the case here. So let's take a moment to admire how awesome these guys are. You know, mimicking not only the colors, but the textures and shapes of leaves. You got holes in there, sort of one that's starting to change colors. Amazing stuff. Okay, so that's great, but what about within genomes? So it turns out there's pretty strong evidence that selection shapes patterns of diversity across the genome as well. And I think the most convincing piece of evidence for this is the correlation of recombination rates with patterns of with levels of diversity. So this was first shown by Dave Gunn and Chip Quadro in the early 90s. So here just looking at the recombination rate or coefficient of exchange and nucleotide diversity, or pi. So selection can cause this in a few ways. One is through selective sweeps, which a few people in this session have talked about previously, but just a quick refresher. So if we have a beneficial mutation here, which I'm showing as a red dot, and over time it's going to increase in frequency fairly rapidly until eventually it's sort of taken over. So it's now fixed or ubiquitous within our population sample, and there's no diversity at the selected site. And as we move out, mutation to some extent, and especially recombination, will rescue diversity from this sweep effect. So we started out with 11 distinct haplotypes here, and then we finished up with 6. And the only reason we didn't finish up with just one distinct haplotype is because of recombination and mutation to a lesser extent. So this creates the sort of signature pattern of a valley of diversity around the selected site. Diversity is eliminated at the target of selection, and as you move further and further away, we recover back to sort of our baseline. This was first shown by John Mayer Smith in 1974. So positive selection creates a correlation between recombination rates and diversity. So negative selection does this as well, it turns out, and this is the phenomenon called background selection, where negative selection reduces diversity of linked neutral alleles. So again, a similar thing is going on, and that we're losing the number of distinct haplotypes. Here our red mutations are deleterious, so they're going to be removed, and when they're removed, their entire chromosome, if there's no recombination, is removed as well, and is then replaced by one of the surviving chromosomes that did not have a deleterious mutation. So we lose diversity through negative selection as well. They're going to call this background selection, or BGS for short. Okay, so both of these models conveniently can produce a very similar sort of V-shaped pattern when we're looking at levels of nucleotide diversity. But we get this valley around a sweep, and we can get a similar thing produced by background selection. And there are nice analytical models for predicting the shapes of these sorts of decays. That's great, but that's just telling us about the levels of diversity. But selection doesn't just affect the amount of diversity, it also skews some of the other patterns. For example, allele frequencies. If we look around a selective sweep, we'll see an excess of both low and high frequency mutations. So this blue mutation here is a singleton, and this gray one is a very high frequency, and that gray mutation reached high frequency because of recombination, rescuing it from the sweep. It didn't quite make it to the fixation there. And this effect will also create an excess of linkage disequilibrium. So these gray mutations here, they show up together, or they're absent together, and that's produced by the hitchhiking effect as well. So we get a reduction in diversity, a skew in the site frequency spectrum, and an increase in linkage disequilibrium. So you have multiple facets of variation are affected by positive selection. Background selection may affect these things as well. So how do we deal with this? Well, it's actually kind of difficult because background selection is a very flexible model. The nice valley in diversity I showed you earlier is produced by something like this, where you have a sort of long chromosome that is missing any sort of functional DNA here, and then we've got our selected sites, our gene, if you will, right in the middle. So diversity is removed from the center, and then recombination eventually rescues it as you move further out. But in an actual genome, selected sites aren't always going to be arranged this way spatially. You may get things that look like this, or this, or this. So what is sort of the expected pattern of diversity in the presence of background selection in an actual genome? And that's what I'm addressing today via simulation. So I'm going to do this by first simulating two different scenarios of BGS. One is sort of that simplistic gene desert model with a gene right in the center of our desert, our little oasis here. I'm calling this central BGS. And the other model is going to be based off of the human genome annotation. So we're going to draw the locations of exons and conserved non-coding elements from the human genome, for example, from the UCSC genome browser here. And we're going to simulate selected sites in these locations. And I'm going to call this real BGS because it's a slightly more realistic model, I think. Okay, so just a few notes on how we're going to do these simulations. So for each replicate, we're going to pick a random 1 megabase segment of the genome. We're going to take it through combination map. We're going to take the locations of coding sequences and conserved non-coding elements, and we're going to throw down deleterious mutations at those locations according to a distribution of fitness effects that was estimated previously from human data. And once we've done the simulation, we're going to repeat for another 1 megabase chunk that we're going to select randomly, and we're going to do this thousands of times using forward simulation. So here's what we see when we do this. Here are the average values across our 1 megabase chromosome of 3 different estimators of theta. We have pi here, Watterson's theta, and theta h from theta moves h, the left-hand side of that one. So what we see here is that we again get this valley of diversity, and we start recovering with increasing distance, and then we sort of get back to our neutral baseline here, which I'm showing as the dashed lines. So that's the average of 1,000 simulations. And here's the average of 1,000 across our real BGS scenario. Okay, this is kind of silly because each replicate has coding elements in different locations. So of course, spatially, no pattern comes out. It all sort of gets muddled together, but you do see that on average diversity is reduced. So it might be more useful to take a closer look at some of these particular regions. So let's take 4 different random 1 megabase chunks, and for simplicity, we're just going to plot pi here. So now for each locus, for each region, I'm simulating 1,000 replicates and showing the average value of pi across a bunch of different windows. So this is not one realization. This is sort of like the expected pattern of pi across this region. And here in this little stack plot at the bottom, I'm showing the density of conserved elements. These are fast cons elements here, and these are coding sequence. So we can see there's sort of like a dip here in the number of selected sites, and we get a slight increase here in diversity. Okay, so these are just 4 randomly selected examples. You can take my word for it. This is the random number generator pick these out. I didn't cherry pick these. We can see that in these 4 examples, we aren't seeing anything like a valley in diversity here. We're seeing more of a straight line. So that may suggest that in actual genomes, BGS is not going to create something that looks like a selective sweep, but we should take a closer look. Just looking at pi there, but there are many statistics that describe all aspects of diversity, and we can look at all of these. This is information overload. You don't have to look at every panel here. My point is just to show that we can and should be looking at not just pi, but also the site frequency spectrum, the amount of linkage equilibrium, which is down here, haplotype diversity, all these different things. So it would be nice if we could use all of this information to tell us something about how often BGS looks like selective sweeps. And we're going to do that via classification. So we're going to use a machine learning method called SHIC, which I developed. That's why I'm using it, because it's easy for me to use, because I've made the damn thing. But the idea here is that it just sort of captures all of that information that I was just showing you, and it does it in a sort of a spatial manner. So each of these statistics is measured in a number of sub-windows around the focal window that we wish to classify. So we're going to determine whether this is a sweep based on the values of statistics in windows surrounding it. So we get this sort of full picture there. So again, we're trying to answer this question of how often do sort of the patterns of diversity produced by BGS look like a sweep? And we were answering that by rephrasing it as another question: how often does our classifier SCHC mistake a region simulated under BGS for a selective sweep? And there are a number of other methods that use similar information, including this recent one called Trendsetter. These can all be used as well. I'm just using SCHC because it's my thing. Okay, so let's come back to our human simulations with BGS here. So if we apply our classifier to a bunch of these simulated regions, in the absence of natural selection, we see that a decent chunk of these are misclassified as a sweep, mostly as a soft selective sweep on standing variation, because those are sort of harder to disentangle from neutrality. So this is our false positive rate there. If we simulate under our central BGS model, then half of all of these simulations are misclassified as a sweep, again, mostly as a soft sweep. So that seems pretty bad, but if we switch to our real BGS model, where we're drawing locations of selected sites from the genome, then we see no significant difference between the neutral case and the BGS case. So here, it doesn't seem that BGS is fooling us into thinking that there are patterns of consistent with selective sweeps. But this is under constant population size, and population sizes are certainly not constant in humans. There have been bottlenecks, especially in out-of-active populations, and these can produce patterns that are somewhat reminiscent of a sweep, where you get rapid changes in allele frequencies. So we repeat this under a European demographic model, and we get a slightly higher false positive rate, which is not surprising here, but it's not any higher for our BGS than our neutral scenario. Okay, so in humans, it's looking like we don't have a real problem here with discriminating between BGS and hitchhiking. Okay, how about Drosophila? We're going to switch to a different genome, different organism here for two reasons: to see what happens when the genome is denser, more densely packed with functional elements in Drosophila, rather than about 5% of the genome being conserved. It's greater than 50%, and there's also a larger effective population size, which will affect the efficacy and the strength of natural selection. So now we're going to look at a few random regions again, just looking at pi, and we can see that the density of conserved elements is much higher here. So negative selection is having a much larger effect on the isodiversity here. When we see a dip, it corresponds with a spike in diversity, and vice versa. So if we see spikes in the density of conserved elements, we see dips in diversity. So we get that nice sort of correlation there. Maybe this means that we're going to have more false positives produced by BGS, but no. The result is the same. You just don't get an appreciable number of BGS examples that look like sweeps, no difference than what you get under neutrality anyway. Okay, so just to quickly wrap up, probably the biggest take home of my study, I think, is that we can and should model background selection via forward simulation in a way that actually matches what BGS will look like in real genomes, where we don't have some central region that's under selection and then a desert of nothingness. We know where these selected sites are, so we should use that information when we're thinking about what these forces do in nature. And if we do that, we see that BGS will not mimic sweeps in actual genomes, and we should be far more concerned with demography than BGS in the context of detecting sweeps. And I think these two forces can be disentangled if we take this information into account. And also, I think we should use more than just pi in doing this. We should use the whole spectrum of diversity. So I'm over, session's over. No time for questions, but yeah, come bug me afterwards.